Why are animals, plants, and fungi so radically different from each other? Do they share anything in common? Yes. They all contain cells with the following common characteristics. The cellular contents are enveloped within a membrane. The cells contain bacteria-sized mitochondria. They contain a nucleus surrounded by a double membrane. They may replicate and divide using a process called mitosis. Here we show the duplicated chromosomes, which are connected to the centrosome by way of spindle fibers, which are also connected to the cleavage site, where the daughter cells will be separating. Fungal and plant mitoses are different from animals, but the results are the same. Characteristics not shared by animals, plants, and fungi. Plants and fungi have cell walls that cover their cell membranes. Animals have no cell wall. Plants generate their own food using a bacteria-sized chloroplast. Animals and fungi have no chloroplast. Plants and fungi only absorb nutrients. Animals can absorb nutrients and ingest larger particles by enveloping them. Animals like humans have hundreds of cell types. Plants and fungi have only a handful of cell types. Let me repeat that. Animals like humans have hundreds of cell types. Plants and fungi have only a handful of cell types. How did that ever happen? Hold the phone. I thought you said animals, plants, and fungi were related. More on that later. If animals, plants, and fungi shared a common ancestor, how did this ancestor make a living? The first true ancestor of animals, plants, and fungi must have been capable of engulfing bacteria as prey, using a flagellum to pull them in through the cytostome or mouth of the cell, into the cytopharynx, the gullet, via the centrosome, and this cell had to have had some sort of nascent nucleus. Here we show the prey bacterium attaching to the end of the flagellum, or the flagellum could just scoop it up and put it into the gullet. In either case, it becomes phagocytized and enveloped by the cell membrane of the host to create a double membrane structure called a phagosome. The phagosome meets up with a lysosome, which is a bag of digestive enzymes. The phagosome and lysosome fuse, allowing the enzymes direct access to the prey. Fusion of these two compartments is called a phagolysosome. The bacterial cell wall and membrane break down, allowing other enzymes to digest the cell contents. The digested contents are spread throughout the phagolysosome. The phagolysosome releases its contents either into the cell as food or ejects it from the cell. Wait just a minute. This sounds like an animal. Plants and fungi don't eat stuff this way. I just can't get anything over on you, can I? Sometimes the lysosome does not fuse with the phagosome. This can lead to the acquisition of bacterial endosymbionts. An endosymbiont is a living organism that lives inside of another organism inside its cells, not just its body. And they coexist together, and they actually work together to create a new kind of an organism. It starts out with another phagosome, but if this bacteria survives, it can replicate in tune with the host and can be passed down from generation to generation. The bacterial cell wall is no longer required in this protected environment and may be detrimental. Such a process may have led to the incorporation of aerobic bacteria, precursors to mitochondria. Photosynthetic bacteria may have been incorporated in the same way. Such photosynthetic bacteria may have been precursors to chloroplast. So far, so good. Now, here comes the controversial stuff. Other bacterial endosymbionts may have found their way into the nucleus. I don't think you'll be finding examples of that in your textbooks, where they are captured by the mitotic spindle and replicated as individual chromosomes. In this diagram, 1X is the total number of chromosomes identical in gene number. Over time, the nuclear phagosome may fuse with the nucleus. This allows intimate contact between the two chromosomes. More nuclear endosymbionts may be acquired, adding still more chromosomes. This process of chromosome addition shown here is linear. Let me introduce a much faster way of getting chromosome numbers up to speed. I call this process hierarchical endosymbiosis. Now I know that's a mouthful, and I will break it down for you as we proceed further through the video. The small cell on the right is the same size as the one shown earlier. It acquired its 3X chromosomes in the same way as before. So did the larger cell. However, instead of taking up bacterial prey as before, the larger cell consumes the smaller one, and the chromosome number doubles. Previously, two separate events of bacterial uptake were required to increase the chromosome number from 1X to 3X. Hierarchical endosymbiosis has allowed a doubling to 6X in just one step. Let's compare the two processes here. To be fair, let's assume both host cells start out with only 1X chromosome. The red arrows in both of these charts are pointing to the nucleus that will be inherited via endosymbiosis by the 1X method versus the 3X method. Now in the 1X method on the left, what's going to happen is that it's going to take up one bacterial endosymbiont per endosymbiotic event, leading to a 1X increase each time an endosymbiotic event occurs. On the right, it's going to be taking up 3 times that amount because the endosymbiont that it swallows has 3 times the number of bacterial endosymbionts than what the 1X method is able to take up. Because this cell is so much bigger, it's taking up a cell that's the same size as the one that we're showing in the 1X chart. Okay, in endosymbiotic event 1, the 1X method has taken up 1 additional bacterial endosymbiont, whereas the 3X method has taken up the equivalent of 3 of them in one fell swoop, so that the chart on the left has 2 and the chart on the right now has 4. In endosymbiotic event 2, the chart on the left has taken up another endosymbiote so that it now has 3. But the 3X has taken up 3 additional, which makes it a total of 7 X chromosomes. Endosymbiotic event 3, the 1X has 4 now, and the 3X has a total of 10. Now imagine a series of hierarchical endosymbiotic events based on a geometric progression of powers of 2. And what do I mean by powers of 2? It's doubling. It doubles each time, like 2, 4, 8, and 16. The slide I'm going to show you next will illustrate what I'm talking about. Okay, that represents one bacterial endosymbiont, has 1X chromosomes, and we are at event 0. It hasn't been taken up yet by another cell. Okay, in event 1, you have your original nucleus, and then it has been added to another one equal in size from a single endosymbiotic event. So the chromosome number has doubled here. But instead of just taking up another 1X chromosome, this next endosymbiotic event is hierarchical endosymbiosis, and it takes up twice the amount that it had before. In event 3, again we take up twice the amount that we had before, and now we have an 8X chromosome number. We continue the process to get 16X chromosomes, then 32X chromosomes, 64X chromosomes, and 128 chromosomes. We could have continued, but we would have run out of space on the slide. So what happens if we carry this progression out to 20 events? Event 8 equals 256 chromosomes. 9 is 512 chromosomes. 10 is 1024 chromosomes. 11 is 2048 chromosomes. On and on and so forth. From 1 chromosome to over 1 million in only 20 events. This is the power of geometric progression over linear progressions. Linear event, 20 equals 20. Geometric event, 20 equals over 1 million, using only powers of 2. Here is a cellular illustration of hierarchical endosymbiosis. So we have the original host cell has 2X chromosomes, and it hasn't swallowed anything yet. And we will be doing this one by powers of 2 also. So in event 1, it swallows 2 bacterial endosymbionts as before, and it increases its chromosome value from 2 to 4X. In event 2, a larger cell swallows 2 of those smaller cells that have 4X chromosomes and increases its value to at least 8X. Of course, it already has some nuclear material to begin with, but we're going to just ignore that for now for the sake of simplicity. In event 3, 2 of those 8X cells are being swallowed by a much larger host cell to give it a minimum of 16X chromosomes. In event 4, we're running out of slide space again, but we're having a 32x host is swallowing two 16x endosymbionts to give it a minimum of at least 32x chromosomes. Now those red arrows are pointing to whatever mechanism was used to draw these endosymbionts into the larger host cell. But in doing so, they used their own centrosomes. So there's a lot of centrosomes involved here, and you may be looking at submitotic events that work together in harmony to produce a single mitotic event involving all of these chromosomes, even though they have their own centrosomes. Evolution always leaves us with signposts to follow. Euglena is one example. Euglena is a single-celled organism that is sort of a missing link between plants and animals. It has a chloroplast. It has a flagella that it can use to move around with, but it can also lose its chloroplast and become phagocytic, use its flagella to eat bacteria. It is at this juncture that the animals, plants, and fungi begin their great schism. As you can see from this diagram, fungi and animals are more closely related to each other than either one of them is to plants. Plants and fungi develop cell walls to counter osmotic effects from water intake. Plants and fungi with no cell walls would have no protection from osmosis in their natural environment. They would swell up with water and burst. By retaining cell walls, however, they lost the capacity for engulfing prey bacteria. One-celled organisms like Euglena have contractile vacuoles that use energy to expel excess water. Multicellular animals have sophisticated cell membrane channels, and these regulate water transport into or out of the cells as needed. So how does this explain why animals have so many different kinds of cells compared to plants and fungi? The ancestors of animal cells retain the capacity for acquiring nuclear endosymbiotes because they have no cell wall. Why would this be important in the evolution of complex animals? It allowed animal ancestral cells to engulf much larger, more genetically complex endosymbionts. This allowed the host cells to sort through all the different types of endosymbiotic cells available to them and retain those which provided them with a selective advantage. Examples of selective advantages: cells that make extracellular connective tissue, can calcify that extracellular connective tissue, make muscle tissue, absorb large amounts of oxygen, digest a large variety of food sources, detoxify a large variety of toxic substances, communicate over long distances within a network, combat foreign invaders, etc., etc. Without hierarchical endosymbiosis coupled with natural selection, this process would have taken forever and a day. Nature always takes the quickest path to success. Natural selection demands it. Hierarchical endosymbiosis is that path. All others just get left in the dust. As I stated before, evolution always leaves signposts. These signposts are fossils, relics of a bygone age. but they also include living fossils. Examples: euglena, jellyfish, cockroach, coelacanth, a fish, salamander, lizard, bird, platypus, an egg-laying mammal, possum, a marsupial, lemur, a placental primate. Evolutionary signposts point the way from the very first bacteria all the way to human beings. However, the elephant in the room has yet to be addressed here. What was the selection pressure that compelled early predatory cells to acquire so many different kinds of genetic attributes? Simply put, it was the filling of a vast, vacant ecological niche. Predators who have the following characteristics win out over those that don't: bigger, stronger, faster, more agile, more resilient, omnivorous, more intelligent. In the most generalized sense of the word, predator refers to the ability to seek out prey and consume it for food. Since this involves planned coordinated movements, it would also pertain to herbivores. Multicellularity coupled with specialized cell types is how all of this can be achieved. And it all starts with a single cell, with all the genetic components necessary to generate a complex multicellular animal. How do all these endosymbiotes determine when to manifest their characteristics? To generate an animal that is more than the sum of its parts. This requires a detailed understanding of how DNA within this first cell joins with other molecules like proteins, so it can be modified and regulated to generate every cell in the body. I believe this has something to do with DNA superstructural changes that must occur. This allows embryonic cells to convert into stem cells, which are in turn converted into adult cells. These cells work in harmony using sophisticated layers of controls. Such a complex system is prone to breakdown over time. Cancer is a prime example of such a breakdown. This is a photomicrograph of a self-destructing cancer cell. It looks like endosymbiotes escaping through channels. Could endosymbiotic chromosomes be attached to endosymbiotic centrosomes in a hierarchical fashion? During cancer, could unregulated submitoses be taking place? Partial replications that corrupt the cell cycle as evidenced by multiple centrosomes? Taking over the cell machinery and forcing it to replicate certain subcomponents in an ever-accelerating fashion? You can learn more about DNA superstructure by visiting other videos associated with this one and by visiting evolution4.wordpress.com. My BitChute channel, shown here on this slide, contains 11 other videos, including 3 that talk specifically about cancer.